Okay, good morning. So let's start off today by discussing the exam that's coming up. So our first exam is this Friday. You can see here on the calendar, this is where we're at. So today is the last lecture of new material. And then so that wraps up chapter 6. So right after the lecture today, I will make the quiz for this material available. And I'll give you some extra time to take it. So the quiz will be due Thursday night by 11:55 midnight. Okay, that way, I think it's important that you try to take the quiz before the exam, because obviously the quiz helps you with making sure that you understand the material that will be on the exam. So don't forget to do that, though, as you're busy sort of caught up studying for the exam itself. So the exam is going to cover all the lectures that we've had so far, including today's, so lecture 10. And then on Wednesday, we will use our class time to review. So the format of the exam is that there will be 25 multiple choice questions, pick the best response A through D, and that's going to be just like the quiz format in terms of the types of questions, the level of difficulty, etc. And then you will also have free response questions. You will have 3 short answer free response questions. So these can be answered, in maybe like 5 or 6 sentences. I'll give you 4 topics to choose from, and you will pick 3 out of the 4 that you want to write about. So those will be your 3 short answers. And then you will have 1 longer answer one. So this is more like, paragraph length type of response. I will give you 2 topics to choose from, and then you pick which of the 2 topics you prefer. Right. So altogether, you'll end up 25 multiple choice. You'll answer 3 short answer. You'll answer 1 long answer during our class time, all right, the 50 minutes. Right. So you'll be doing this just like you're joining class like normal. So you'll log on with Zoom and you'll have your cameras turned on so that myself and Alexis and Zoe can help see you and proctor the exam with you. And then when everybody's kind of set up and ready to go, then the exam will become available in La Lima. And then you just open La Lima and you go ahead and you work on your exam, right, during class. Right? So it's important then that you do have a working webcam. If the webcam on your laptop is busted, one thing that you can do, students have successfully done, is to do the Zoom part through your phone, right? So log into Zoom for the class over the phone, and you just set your phone up somewhere next to you on your desk or whatever, so that the TAs or TIs can see you, and then you go ahead and you do your La Lima test. Okay. All right. So if you have any questions about that, feel free to shoot me an email. But I think it's pretty straightforward. And I think this is, the format of the mix of multiple choice and free response is how I've always done this class, because I think it's really the best way to, first of all, cover a lot of material through the multiple choice, but also through the free response, give you a chance to really kind of express your understanding of certain things that wouldn't necessarily come across well just by multiple choice questions. Okay. All right. So let's continue where we left off last time. Last time we were looking at the activation of RAS, and we said that RAS is involved in 3 sort of main pathways that are relevant to the development of cancer. And the pathway we looked at last time was the one that's in the middle here. This is RAS activating a series of different protein kinases. These kinases, when they're activated, they phosphorylate another product.
I found this on the web for series of different protein kinases.
And that's why you have eventually, I said a series of protein kinases, then Siri on my phone woke up and went ahead and found a whole bunch of websites, I guess, about MapKs. But anyhow, so remember, these are kinases, a string of kinases that work upon each other, eventually resulting in the activation of certain transcription factors that will then turn on different genes, etc. All right. So that's where we left off last time. This pathway over here, the RAL GEFs, we're not really going to go into because we don't have enough time for that. But the one on the left here, this is the third RAS pathway. This is one that's very important, and a lot of cancer research is focused on this. And this has to do with RAS initiating the PI3 kinase AKT pathway. These little illustrations on the top, these are kind of nice, right? Because these are actually the 3D ribbon structure of RAS in green with the sort of signaling domain of it in purple. And then you see the molecules underneath it are the various partners that then participate, right? RAS communicating with them to activate things like, so this light blue one might be RAF, for example, right? To start off the RAF MAPK pathway. This one over here is PI3 kinase, PI3K, this orange one. Right? So to this date, we actually don't have a very detailed understanding of how RAS actually activates these downstream partners, because RAS itself is not a kinase, right? RAS is kind of limited to just being a G protein, right? So its job is to bind guanosine nucleotides. So in this case, right, it binds GTP. So that's what this little dark blue navy thing in here is. This is GTP. And the binding with GTP causes RAS to change its shape, and then it can associate or communicate with other proteins. Right? Remember, when RAS normally is bound to GDP, right, guanosine diphosphate nucleotide, its shape is in the off position. Right? But when it binds GTP, right, it's in the on position, it communicates with these other proteins. And so I think we believe now that really what it's doing is it's either stabilizing these proteins in a conformation that's an active conformation for that protein. So otherwise, things like the RAF might just always kind of fluidly be in between its sort of active and inactive conformations. So if you can stabilize the active conformation, that's how RAF signaling can happen. With PI3 kinase, we believe that the job of RAS is more to actually help PI3 kinase be held in a particular location in the cell, right underneath the cell membrane, where all of this stuff is going on. And that encourages PI3 kinase to be activated through associations with things like the big epidermal growth factor receptors, right? The big like tyrosine kinase receptors. Right? So PI3 kinase works through AKT, right? And you can see the activation of AKT results in these very critical, pretty obvious, important phenotypic changes that are related to cancer. For example, activation of AKT can inhibit apoptosis from happening, right? So this keeps cells surviving, right? They don't self-destruct. It can also activate proteins such as mTOR, right? So I think a couple of you had article share papers that you chose that talked about proteins like mTOR that stimulate protein synthesis, cell growth. And AKT can function through things like the GSK3 beta pathway to stimulate cell proliferation. Right? So we will look at some examples of these as we go forward. But first, let's talk about how this PI3 kinase activates AKT, right? And why it is that this idea of RAS bringing PI3 kinase to the inside of the cell membrane is so important. So here, this is an illustration of our cell membrane. So remember, our cell membrane is 2 layers of phospholipids, right? So these big gray lines are 2 layers. We're talking about the layer that's more sort of the inner layer here. And you recall it's made up of these phospholipids. So you'd have a phosphate group attached to a glycerol, right? And the glycerol kind of acts to hold these 2 sort of fatty acid chains together, right? So this is the sort of basic component of a membrane phospholipid. Normally, you would have some other type of functional group here stuck to the phosphate group. The majority of times, the molecules that make up our phospholipid bilayer, you would find a choline group attached here, right? But all kinds of groups can be attached to the phosphate group. So in this particular case, a number, right, it's quite common that you would find these inositol groups attached to the phospholipid. Right? And so this is an inositol. It's a ring here. So you can think of it as like a ringed alcohol. Right? And so we call this phosphatidyl inositol when it's bound to the phosphate group here as part of the phospholipid. Right? Certain kinases, so phosphatidyl inositol kinases, can come and add additional phosphate groups to it. So here you can see this PI kinase has come along and added 2 additional phosphate groups at the 4 and 5 position, changing this from a regular just PI to now a diphosphate, PIP2, right? 2 phosphate groups attached to the PI. Okay. So oftentimes, this PIP2 can serve as a signaling molecule inside the cell for various functions that aren't necessarily super relevant to cancer. The way that works is you have an enzyme called phospholipase C, or PLC. You might remember, oh, PLC, that's kind of familiar. Did we look at that? Yeah, that was one of the examples of proteins on a previous lecture slide where we looked at how many proteins are just made up of different types of Sark homology domains, or SH domains, like SH2, SH3. Remember, and those types of domains, especially things like the SH2 and SH3s, are in charge of oftentimes binding to things like phosphorylated groups. Right? So phospholipase C will go ahead and bind to this, and it'll cleave. It'll cleave the PIP2 off of here, right? And then this floats away, and this is just called inositol triphosphate, or IP3. And this can be used as a signaling molecule inside the cell. However, right, it may also be that before the PLC gets there to do it, you have another protein, and this is the one we're talking about, PI3 kinase, right? That takes a PIP2, right? And if you follow this arrow over here, around to here, it adds a 3rd phosphate group on it. So now the PIP2 becomes PIP3, right? And this is very important because this PIP3 is now the right shape to activate another protein, AKT. Right? So let's look at this here. Here we have the PIP2, right? And before the PIP2 got cut off by the PLC, a PI3 kinase had a chance to work on it, and the PI3 kinase put a 3rd phosphate group on the ring, turning it into PIP3. And now this PIP3 form is the right shape for this AKT protein that's floating by to now bind to it. Right? And as a result, this AKT might have been happily just floating around in the cytoplasm somewhere, minding its own business. Now it gets stuck to the underside of the cell membrane, right? Because of this PIP3 signal. Once it's there, there are other proteins that will happily work on the AKT, right? So in this case, you have these PDKs, these phosphoinositide-dependent kinases, right? So they're kinases, so they will phosphorylate some target protein, and they're dependent upon these phosphoinositides, right? So the fact that you have to have these formations of things like PIP3 allows these things to go to work. And in this case, because the AKT is stuck there right next to where these PDKs are by the cell membrane, right? Their favorite target then is the AKT, and you get phosphorylated AKT, or phospho-AKT, that is the activated form of AKT, and now starts kicking off those big pathways that we're talking about. For example, inhibiting apoptosis from happening, or increasing protein production as part of cell growing, or stimulating proliferation. So you can see why this PI3 kinase phospho-AKT pathway, if something goes wrong here, right, it could be a real key player in driving transformation of a cell, right? And if we trace this back, let's think about this, right? This all had to do with PI3 kinase being allowed to preferentially go to work on the PIP2, right, turning it into PIP3, and that PI3 kinase was brought here by RAS, right? RAS that brought the PI3 kinase here when RAS is active, right? And how did RAS get active? through something like one of those receptor tyrosine kinases activating RAS. So now PI3 is brought here also by RAS. So PI3 kinase can be activated by the RTK or the receptor itself. And then PI3 kinase goes to work, making these PIP3s that then activate the phospho-AKT, right? So it's kind of, I mean, I know it's confusing, but at the same time, it's wonderful because you see how it kind of comes together, right? And how these things are related. So very importantly, right, if the activation of AKT through phosphorylation is going to start driving all these potentially oncogenic pathways to happen, then you have to imagine, hey, this is an important opportunity here for the cell to control this, right? And make sure that you don't have things like phospho-AKT forming out of control. So how can our cell do this? Well, one of the best ways is to get rid of this PIP3 signal before it has a chance to activate the AKT by bringing it here, right? Because without the PIP3, the AKT might just float by. So how do we do that? There is an important protein known as PTEN. PTEN does the opposite of PI3 kinase, right? So PI3 kinase added the third phosphate group on here, making PIP3. PTEN reverses it. It takes that third phosphate group off, converting it back to PIP2, right? Therefore, you can say the function, the eventual downstream significance of PTEN is to stop oncogenic pathways. So instead of encouraging tumors to happen, PTEN is the opposite. It is a quintessential example of a tumor suppressor protein, right? And we will study many of these tumor suppressors, and we'll begin to appreciate that just as important as it is to study oncogenes, proto-oncogenes, and how they can become oncogenes, right? And how that can happen through mutations to lead to cancer. Also, just as important is understanding the normal role of tumor suppressor proteins like PTEN and how their disruption due to mutations can also play a critical role in the formation of cancer. So this is an amazing slide here. I love this, right? So on the right, we have some images, histological images of some pathology slide where they've taken a section of a patient's biopsy of a tumor region. So this is some sort of glandular epithelial tumor that's forming here. You can see this is the epithelial layer, and then there'd be like a basement membrane underneath it, and then this will be the stromal or mesenchymal layer, right? And these two types of cells, the mesenchymal stromal cells and the epithelial cells, normally be talking to each other. Remember, the epithelial cells normally should be very well behaved, right? But look, this section of the tissue, the lining of this duct or whatever this is, you can see the transformation of the cells happening, right? They're beginning to get really misshapen. They're beginning to really become larger, proliferate, right? And what they've done, the nice thing about this section of the slide is they've made sure to capture some of the neighboring epithelial cells that border the tumor here. And they've stained this, right, for the presence of normal functional PTEN. So this on the left is just a little recap of the previous slide. Remember, what's the job of PTEN? PTEN's job is to reverse the formation of PIP3, right? The PIP3 that's being made by PI3 kinase that's going to drive activation of AKT. PTEN quickly reverses that, bringing the PIP3 back to PIP2 so that the AKT cannot get eventually phosphorylated, right? So they're looking at the staining here of levels of PTEN. So in these cells that are beginning to transform, right, what they're saying is, your cells have two copies of PTEN, one that came from mom, one that came from dad, right? And if those copies are wild type or they're working properly, you can call them sort of your positive or plus, right? The working version of that gene. So already in these cells on the left, they've lost one copy, right? Possibly through mutation, right? But because they still have another good wild type copy, they're still producing a lot of PTEN. And that's why the brown antibody stain here for the PTEN protein looks really dark, right? But look at the PTEN status of these cells that have now really started transforming. They've lost that other good copy of the PTEN, right? And you might say, but then how come they still stain brown? Remember, when we say they've lost it, that doesn't necessarily mean that the mutation has led to zero production of PTEN protein, right? This could be a mutation somewhere in the coding sequence of the PTEN so that the version of PTEN that's made is defective, right? It doesn't fold up properly, or it's truncated in such a way. So maybe the antibodies can still kind of pick up on it, right? But very clearly, these cells are not making nearly as much PTEN as the normal wild type here. So this is the best part of this slide. What if you looked at those same cells? So this is just, when you're preparing histology slides, you'd have a tumor mass or a section of tissue that they embed in paraffin, right? So a block of basically like hardened wax. And then they can slice sections, super thin sections of that chunk of paraffin wax that has the protein embedded in it. So you get these very sort of few micron thin slices of the tumor, right? And what's really awesome about that is you can take two of these slices that were right next to each other, like here, and you essentially get two sets or two samples of the same group of cells that you're looking at, right? And so you can stain this one slide with antibody for PTEN to look at the change in PTEN levels. The very next neighboring slide, you can stain for phosphorylated AKT, right? So here the brown is showing you where you have phosphorylated or activated AKT, right? Wow, look at this. It's like night and day, right? When you still have PTEN functioning, you have low levels of phospho-AKT. Once you've lost functional PTEN, the phospho-AKT goes shooting up, right? And that certainly could be part of what's driving the transformation of these cells on the right. Okay, so really neat example to get you guys thinking about what you're looking at here. All right, so this kind of summarizes what we were just talking about. So here we have, what's the best way to talk about this here? All right, so right now we're talking about PI3 kinase activation, right? So PI3 kinase, maybe it would be floating around somewhere in the cell normally, but if it gets brought to the cell membrane, it has an opportunity to convert PIP2 into PIP3, right? And if that PIP2 gets converted into PIP3, then AKT, so this blue thing here, right, can now bind to PIP3, and waiting for the AKT already at the membrane are these phosphoinositide dependent kinases like PDK1, PDK2 that happily then go to work on AKT because it's being brought there, right? And that would then phosphorylate the AKT, and that would then drive certain pathways like cell survival, right? Anti-apoptotic pathways. So let's back up. How did the PI3 kinase even get brought here in the first place? Because it was floating around somewhere here as well, right? It was brought here because RAS brought it here. RAS, that's in its GTP-bound activated state, right, then interacts with PI3 kinase, bringing the PI3 kinase here so that it has an opportunity to be activated by a receptor tyrosine kinase, so it can make these PIP3s. How did the RAS get activated? Oh, by that receptor tyrosine kinase itself, right? Working through those different types of adapter proteins like the GRAB2 and SOS that start interacting with the phosphorylated tyrosines, and they then help drive the exchange of RAS giving up its GDP and getting a GTP instead, thereby activating it, right? So just think, this is all driven by the fact that this orange diamond up here arrived on the outside of the membrane, right? That's the amazing thing. This orange diamond is the small growth factor that arrived from something like the mesenchymal cells underneath the basement membrane that decided to tell this cell here to go ahead and start dividing and proliferating, right? Prior to this growth factor arriving, this receptor tyrosine kinase wasn't even together as two parts. It was kind of floating around as monomers. And all of these other things, right, especially the PI3 kinase and the AKT, they're floating around somewhere else, right? So the chances of them coming together on their own, right, are kind of almost astronomically slim, right? But when the orange diamond arrives and the receptor tyrosine kinase dimerizes, its tails auto transphosphorylate each other. Then you have the activation of RAS taking place, the RAS bringing in the PI3 kinase, the PI3 kinase gets activated by the RTK. The PI3 kinase makes PIP3. AKT then can attach here. What's waiting for the AKT? These PDK proteins that phosphorylate it. Then you've now activated the AKT pathway, right? Really amazing to think that these things just come together because of this one event of a little protein arriving on the outside. And you guys know I always beat this to death in like Bio 275, right? These molecules, they don't have brains. They don't know what they're doing. They're not planning ahead, right? They just do what they do because they happen to be the right shape. So that if something initiates a chain of events of things interacting with each other, you end up having things like really important cellular pathways activated. Okay. So let's look at some other examples then of receptors that are also communicating with the outside that signal through RAS, for example. We mentioned integrins as examples of non-receptor tyrosine kinases that still play an important role in cell proliferation and cell mobility, right? Today, we're looking at the fact that these can oftentimes still involve things like RAS. So in this case, integrins, remember, they interact with the extracellular matrix on the outside, and that changes their shape. And the proteins that then combine to the cytoplasmic ends of things like integrins, they begin to do things like reorganize the cytoskeletal structure. Right? Part of that reorganization is bringing many of the proteins that are kind of hitching a ride on the cytoskeleton together also. Right? So again, that theme of bringing things together so that things can interact with far more probability or likelihood than if they were just left to wander around on themselves. Right, by themselves. So that includes RAS, right? So you can see these integrins working through different proteins like talons, and these are what we call focal adhesion kinases. So remember I showed you a slide last time, a really cool slide of if you stain the cytoskeleton, cytoskeletal proteins of a cell with some fluorescing, like green fluorescing type of marker, and you have the cell on a tissue culture dish, you can actually see where certain proteins of the cytoskeleton are sort of coming together at the points where literally, right, the cell is touching the plastic. So in that slide, it was like green cytoskeleton and these orange, bright little orange spots where the cell was actually touching the surface. So these are proteins like the focal adhesion kinases being brought together there, right? And they are then, what, going to work, doing their kinase function, phosphorylating any protein that kind of gets brought close to them as well. Right? So all these other proteins that are hitching a ride on the cytoskeleton, right, they get phosphorylated. And that's how you also do things like you start recruiting the adapter proteins like GRAB2 and SOS that activate RAS nearby. So here's another picture. I don't remember if this is the picture I showed you last time, but what I wanted to really illustrate is if you have a lot of extracellular matrix proteins, fibrinogens, and different types of things in the extracellular matrix that make, for example, the basement membrane, right? Remember, the basement membrane is a big network, a meshwork of all these types of special protein fibers that the epithelial cell sits on, right? And how does the epithelial cell know that it's sitting on the basement membrane? It's because of interactions of these types of receptors like integrins, right? So these integrins that are on the surface of the epithelial cell, let's pretend this is all the epithelial cell on the bottom, right? Has these different types of extra or ECM sensing receptors on it. When they attach to the different types of proteins and things that they're the right shape to, then that part of the membrane would have a lot of these receptors just kind of being stuck or encouraged to come together because they're all stuck to this little patch of the basement membrane here, right? So what's going on inside of the cell? Inside of the cell, you have the cytoskeletal matrix here, or network of all kinds of things like actin filaments, right? And all these proteins that are all kind of hitching rides on the cytoskeleton. So as on the outside, these receptors are being brought together. That's what these, light blue arrows are supposed to show, that we're heading towards the middle of the slide here, right? Because more and more of them are attaching to this little patch of the basement membrane, right? On the inside, because their tails are connected to different proteins of the cytoskeleton, all of these proteins in the slide are also being brought together, right? And that's how all of a sudden, boom, you have all these interactions between things like SARC and RAS, right, that start kicking off these pathways, including PI3 kinase as well, right? PI3 kinase is now being encouraged to come here, right? And then it has an opportunity, for example, to interact with activated RAS, right? And if here, not pictured, but if here you also have EGF receptors, right, then you can really imagine that these things would start really activating. Okay. So this significance of epithelial cells needing to sense that they are properly attached to a basement membrane in order to be happy, or importantly, in order to keep surviving, right? Because remember we said that some of these signals, if they go through RAS and eventually PI3 kinase activating AKT, those are the signals that encourage survival, right? And are anti-apoptosis, right? That depends upon sensing the extracellular membrane, right? So you can see here, during the development of ducts in certain glands in our body, I think this is mammary gland. What we're looking at are cross sections of different ducts that are forming in the mammary tissues, right? So all of this black space would be sort of the mesenchymal cells, right? The connective tissue in the mammary tissue. And then you'd have these ducts. So these are in cross section, right? So this would be like a tube that's forming going in and out of the computer screen here, right? So these tubes, the lining of them are epithelial cells, right? So that's what these blue cells are. And these blue cells, you know, I love this illustration because what's the red thing here? The red thing is the basement membrane, right? That these blue epithelial cells need to be attached to in order to be happy and to survive. So as these ducts form, you know, you may not have really thought about how do ducts like this form? How do tunnels, you know, vessels in your tissues actually form, right? It's not like there's some big machine that like carves a tunnel through the tissues. They actually form as solid cylinders of cells, and then they get hollowed out on the inside, you know, not by a big machine going through, by just those cells that are in the middle dying off, right? Undergoing apoptosis. So what you're left with is a hollow tube. And that's what's happening here. So the developing epithelial cells, they sort of are playing musical chairs. You know that game, musical chairs, where you all run around like a number of chairs, and then when the music stops, you got to be able to find a spot, right? So all of these endothelial cells have to find a spot where they're able to attach to the basement membrane so they get those survival signals that prevent apoptosis from happening. The cells in the middle, right, of this, they no longer get those signals. So their apoptotic pathways activate, right? And they undergo apoptosis and they die off. You can see the bright green here, they're staining for caspases. So caspases are those groups of proteins that actually drive apoptosis to happen, right? So you can see these cells are expressing caspases and they are starting the process of apoptosis, right? And so they die off in the middle, leaving you a hollow duct, right? So that would be the normal behavior here. But look at what's happening in a precancerous set of tissue. Right? You can see the endothelial cells that are no longer attached to the basement membrane, they are not undergoing apoptosis. Right? So this is really bad because it might mean that these epithelial cells now can just float away somewhere, right, and start improperly trying to find a home somewhere else in your body. Right? So that's very important. This process of developing epithelial cells inside of a developing duct undergoing this very specific type of apoptosis, right? I mean, you know, because they're not like sick, they're not bad or damaged. It's just as part of the normal development of the tissue, right? They are not in the right place to survive. So they're told to undergo apoptosis so that you can get a hollow, properly formed duct. This type of apoptosis we call anoechus. It's kind of a weird word, right? But it has to do with when epithelial cells aren't attached to a basement membrane, right? They are told, oh, sorry, right? You better undergo apoptosis. So they undergo anoechus, which is an important evolutionary adaptation to fighting cancer, right? Because otherwise, if your circulation was just filled with all kinds of epithelial cells floating around, attaching in different places, trying to start growing, right? You'd have lung epithelium growing in your heart, right? You'd have brain epithelium growing in your kidneys, right? You can't have that, right? So this is a very important concept. All right, so that brings us back then to our cartoon here of these various pathways that we've been looking at, right? And how they all kind of rely upon the interpretation or the arrival of different signals on the outside of the cell being interpreted by different types of cellular receptors that then kick off the intended sets of signals inside of the cell. Right? Let's look at a couple of other pathways today. Let's start off with the cytokine pathways. Cytokines are small protein signaling molecules that are typically communications between different cells of our immune system, right? Ways of things like our leukocytes, our white blood cells, to communicate with each other. And so cytokines can stimulate the differentiation of things like monocyte cells that are the precursor of our macrophages. They're going through the blood, right? If they receive the right kinds of cytokine signals, then they will leave the blood, go into the tissues that might be the site of infections, for example, and these cytokine messages will stimulate these monocytes to fully differentiate into macrophages and start going to work, fighting infections, right? So that obviously is a signal that's sort of pro cell cycle, proliferation, right? So those can also be very important in understanding different types of hematopoietic cancers. So let's look at cytokines. They signal through what we call this JAK STAT pathway. Right? So JAK STAT, we looked at this briefly last time as an example of different types of receptors that the receptor itself is not a tyrosine kinase, right? But they might work by recruiting the activity of other non-receptor tyrosine kinase kinases, right? So here you have a cytokine receptor, and in the cytoplasmic end, it recruits these tyrosine kinases, JAK and TYK2. Right? Remember, JAK is the sort of the founding member of the Janus kinases. TYK2 is another example of one of these. I think TYK2 literally just stands for tyrosine kinase 2, right? But when a cytokine molecule arrives on the outside, the cytokine receptor is assembled so that JAK and TYK can now transphosphorylate, right, each other, and then eventually their activation then phosphorylates tyrosine residues on the tail of this cytokine receptor, right? So you might get confused. You might say, wait a minute, if this is a membrane receptor that has tyrosines that get phosphorylated, why do we say this is not an RTK? The distinction is, remember, the receptor itself does not have tyrosine kinase domains in it, right? The classic RTK has receptor tyrosine kinase domains as part of its tail, so it can go ahead and autophosphorylate itself, right? In other words, it can carry out tyrosine phosphorylation on its own. In this case, the cytokine receptor doesn't have such domains, but it has domains that recruit kinases that have tyrosine phosphorylation activity, right? And so then they phosphorylate the tyrosines on the tail of the receptor that recruits these STAT proteins, right? So these STAT proteins, you can see here, STAT1, STAT2, they have SH domains in them, SH2 domains, right? So those SH2 domains, remember, they love to bind phosphorylated tyrosines. So it's kind of funny. Look, the STAT2 has a tyrosine in this position and an SH2 domain over here. The STAT1 is like the opposite, an SH2 domain over here and a tyrosine hanging out over here, right? So as a result, when they're brought together, right, they can actually detect each other, right? The phosphorylated tyrosines that are made by TYK2 and JAK1, right, allow the STAT1 and the STAT2 to now come together, right? And this dimer of STAT1, STAT2 acts as a transcription factor. It then goes to the nucleus, binds to certain sequences, like in promoters or enhancers of genes, that then when those genes are turned on and expressed, right, drive forward things like the proliferation, etc., and the survival of the cell. All right, so that was JAK STAT. Another example over here, this funny wiggly looking receptor that's on the surface, right? It's drawn quite a bit different than the RTKs or the cytokine receptors. What's that about? So this is the signaling pathway that goes through, what is it? Beta catenin. All right, so many of you have probably heard of beta catenin. Beta catenin is another very important, potentially oncogenic protein in cancers. All right, so let's look at what's going on here. So this is an illustration of what we call the canonical Wnt Beta Catenin signaling pathway. So anything that's canonical kind of means like what? This is like the main sort of like the main way that people think of the signaling pathway, right? It's like canon, right? But of course, this doesn't mean that there are not alternative ways of activating beta catenin, right? But this is the classic one that people refer to. So this can be a little confusing. Let's start on the right part over here. And let's start looking at this, right? So this blue kind of pill-shaped, lozenge-shaped thing, this is beta catenin. So beta catenin is a transcription factor, right? So this protein, if allowed to do its job, it will go into the nucleus and it will bind to promoters, enhancers, whatever, of genes to help activate them. And in this case, the beta catenin, when it's allowed to go into the nucleus, it binds to this partner known as TCF-LEF, right? And it encourages this TCF-LEF transcription factor to start doing its thing and activating gene transcription at this site, right? The process of activating the TCF-LEF, right, required that the beta catenin booted off this other brown protein that was bound to TCF-LEF. Right? And this other brown protein is what we call Groucho. Right? Yeah, you can tell these are proteins that were identified in Drosophila, right? And those Drosophila scientists, they have that wacky sense of humor here, right? I don't know why it's called Groucho. Or maybe the flies that have a mutation in it are in a bad mood, or they have a funny mustache or something. I don't know, right? But anyhow, the point is normally these genes are shut down because the TCF-LEF is there, but it's inhibited by Groucho on top of it. Right? So therefore, this gene doesn't get expressed. But if beta catenin comes in, it boots off the Groucho and encourages the TCF-LEF to go to work. Right? So how is it that normally beta catenin is not allowed to turn on these genes? It's not allowed simply because beta catenin is rapidly degraded by your cell. Right? So this is a example of how cells can often control the critical function of important signaling proteins like beta catenin simply by just degrading them and eliminating the amount of beta catenin that's floating around in your body. Right? You might say, well, that sounds kind of weird because that implies that your cell is always making beta catenin, right? So that seems wasteful. You're always making beta catenin and you're just destroying it all the time. What's the advantage of that? Right? The advantage of that is that the beta catenin is always, you always have a steady pool of it being made already, so that if you need beta catenin to switch on, right, you have a lot of beta catenin protein just as long as you don't degrade it, right? Rather than saying, we're not going to make the beta catenin protein until we need it. Then if you suddenly need it, oh boy, you got to turn on the beta catenin gene. It's going to take several hours to start making enough beta catenin, right? It's better to have a pool of this protein around that you constantly just degrade, so that if you need it to work, you just don't degrade it, and then the cell and then the protein go to work. So how is it normally degraded? Beta catenin is normally held in this complex of these other proteins that involve these other critical factors: GSK3 beta, right? That is working on the beta catenin, and this APC. This APC is adenomatous polyposis coli. All right, you don't have to remember the name of it right now because we will study this APC later on. But the name adenomatous polyposis coli already gives you a hint as to in what disease this APC gene or this protein was first identified in, right? So polyposis means polyps, adenopolyposis, glandular polyps, coli is the colon, right? So this is originally identified in colon cancer, right? So APC, GSK usually forms this complex, where GSK, it's a kinase, right? It modifies beta catenin, phosphorylating it. You see the phosphate was added to it. That phosphorylated form of beta catenin is rapidly targeted for ubiquitination and destruction, right? So above it, in this figure way up at the cell membrane, you have that funny wiggly receptor that we looked at on the previous slide. This is the frizzled receptor, right? Again, named after, some fly quality, right? Mutant flies that have mutant frizzled. I'm pretty sure they looked frizzled, right? So normally this frizzled receptor is just hanging out here. If you have this signal, this small protein known as Wnt, right? If Wnt comes and binds to this, then we see what's going on here. Wnt activates the frizzled receptor. The frizzled receptor talks to a couple of other proteins here like disheveled, right? And that attracts this big protein, the axin. And the axin normally is a critical part of forming this beta catenin degradation complex. Without the axin, the APC, the GSK3B, they cannot do its job phosphorylating beta catenin. Beta catenin then is allowed to escape being degraded, right? And then the beta catenin goes into the nucleus and activates all these genes that are responsive to beta catenin signal arriving, okay? So if you look at this here, again, here's the complex, this complex that normally degrades beta catenin, right? Beta catenin is phosphorylated because of GSK3 beta activity working on it. The GSK3 beta activity only has a chance to work on it if it's part of this well-formed complex involving axin and APC, right? So if this comes together, beta catenin is degraded. Okay? So look at this here. So this is a Drosophila embryo, so when it still looks like a grain of rice or something like that. And what they're looking for is levels of beta catenin. And you can see the faint white shapes here represent cells in different bands of the developing abdomen of the Drosophila where you find beta catenin. So beta catenin is glowing here, but you can tell it's very tightly controlled and it's only being expressed in those cells that are meant to proliferate and start forming the different abdominal segments of the fly, right? In between, you can see it's dark, right? So those cells in there, right, you don't have beta catenin signaling, right? Probably because in those dark cells, right, you have this degradation pathway going on, right? So that the beta catenin levels don't really build up and are not stabilized. But in the cells that are brighter, those are cells that might be receiving that Wnt signal, right? That Wnt signal on the frizzled receptor that does not allow this complex to form and therefore allows the beta catenin to signal and encourage those parts of the abdomen, those segments to divide. So look at this fly on the right. This fly has a mutation in its GSK3 beta, right? So this GSK3 beta, it has a mutation in it so that it cannot phosphorylate the beta catenin. And as a result, look at the abdomen, right? It has tons of uncontrolled beta catenin signaling, right? Because the beta catenin is not being degraded in the proper pattern, right? So this then leads to this, all of these cells dividing, and obviously this fly is not going to develop properly, right? Okay, so what about in mammals, right? So yeah, so beta catenin also can become deregulated in cancers. So this is a set of liver cells in a mouse that was exposed to different carcinogens. And what they've done is they're now staining these liver cells for beta catenin protein levels. So the normal healthier cells would be the ones on the right and maybe to some extent on the left, right, where you don't get a lot of brown staining, because in these cells, the beta catenin is being turned over and degraded in its normal fashion. But look at these transforming cells in the middle, right? So some mutation or set of mutation that has happened so that now you're not getting the proper degradation of beta catenin, and beta catenin is really building up and signaling out of control. Right? So that brings us back to our analogy of the orchestra. I'm going to kind of wrap up here because I know we're running out of time, right? So we said the orchestra then, when we think about the harmony of all these different musicians having to work together in a coordinated fashion in order to produce the desired harmonious music, right? That's a lot of stuff, a lot of interactions, a lot of communications, and a lot of interpretations of signals. Right? that must all be working properly. So when we think about this as our orchestra then, right, this is the orchestra that we're talking about, right, where all of these are the different musicians, right? These are all the critical players here, right? They're following the genetic instructions that code for them, right? And they're being coordinated by the efforts of things like the conductor, which are these receptors that are interpreting different signals and making sure that all of these pathways in the cell, right, are properly working in harmony. Right? And you can see many of the things that we've talked about, RAS, SARC, PI3 kinase, AKT, right? Things like here's the GSK3 beta and the beta catenin. Here's MYC, right? You begin to really get this impression of how complicated, right? And how nuanced, meaning how subtle shifts are that can take place that ultimately control how your cells express their genes. Right? And so this last slide then is just a thought question. This is just meant to be for fun. How many cells make up your hand? Right? I don't know if you ever thought about this, but our hand is something that we see a lot, right? And we don't see our faces normally unless we look in a mirror, but we see our hands a lot. Have you ever thought about how many cells you're looking at when you look at your hand, right? I mean, just do that right now. Look at your hand and you can guess. Well, we have about 37 trillion cells in our entire body, right? So our mass of our body is often, you can think of it as a function of having 37 trillion cells in it. So the hand is about 0.66% of our total body weight, right? So one of our hands. So if you just kind of roughly think about it that way, that means you have about 200 billion cells in your hand, right? Just looking at your hand right now. And this is the amazing thing to think about, right? In our everyday lives, we don't give a thought to this at all, that every single cell in your hand here, right, at every single moment of your life is responding to the signals that control their proliferation, their differentiation, right, their survival, and whether or not they stay put or move around, right, all based upon these things that we're studying. Right? And the remarkable thing about it is, thank goodness that all of these things happen without us having to think about it, right? It's all a function of how we evolved with our genes that code for these proteins that fit together in these intricate pathways, right? And how wonderfully beautiful that is, and how, in some ways, how scary it is when you realize when things can go wrong in these critical pathways. Okay, so that's where we'll end today. Sorry, I went over. I think the whole online teaching thing makes me go rambling more than I normally do. Okay, I'll stop the